Okay, team, here we are again with our next video. This video is going to cover organic compounds. We might even get into cells, depending upon how far we get, depending on how motivated I am. Oh, someone's got a leaf blower going out there. Here we go. So this next PowerPoint, just so you know, there's the first one we did, basic chemistry. This next one is actually in the next module. It's in the biochemistry module. And what we want to talk about is organic compounds. Let's remind ourselves again what a compound is. By definition, a compound is simply a molecule made up of two or more elements. And organic compounds are carbon-based. Carbon is the building block of living things. And what we're going to see is that all of these different kinds of organic compounds that we're going to talk about in just a second are carbon-based compounds, okay? Let's go ahead and list them real quick. Let's list all four before we get any further. 4 classes of organic compounds. Basically, all living things are made up of these 4 classes of organic compounds: carbohydrates, proteins, lipids, which include fats and waxes and oils, and the last one are the nucleic acids. So you're not going to want to walk into the next test without knowing these 4, and what you want to know about them is what are they made out of, where are they found, and what do they do. So there's a lot to know about these, okay? If you analyzed living things, like let's say you analyzed a human and broke it down into all of its different elements, it turns out that 99% of what we are made of are carbon, hydrogen, oxygen, potassium, sulfur. We're mostly water, right? So H and O you would expect to be in there. And, the other 1%, well, that's all the other stuff, all the micronutrients and minerals and whatnot. So we have, what we're going to do is we're going to see these in pretty much all of them. Oh, I left nitrogen out, I'm sorry. Put an N in there for nitrogen. I thought I was missing something. Okay, there we go. All right, so let's start out with carbohydrates. And there's a lot to talk about with carbohydrates. You've heard the word carbohydrate. You're familiar with some of these different compounds: monosaccharide, disaccharide, and polysaccharide. We want to know what that means. What we want to understand about all carbohydrates is that there is the glucose molecule, which is a ring type molecule. And I'm going to draw glucose right here. These are carbons. There's the carbon here. And then coming off of these carbons are hydrogens and oxygens, hydrogens and hydroxyls. Okay, and so remember what these lines represent. Every time you see a dashed line, for example, between these carbons, that represents a covalent bond. Look at all the covalent bonds that you're looking at here. If you add all these up, you get C6H12O6, which is glucose or a glucose-like monomer. So monosaccharide means one sugar, and a good example of that is glucose, right? It's a single molecule that makes up the sugar. But what you can do is you can stick these glucose rings together, and we're going to see that in just a second. So, glucose is a molecule that you need to be familiar with. We're going to be talking about it a lot. And if we move on to the next slide, we can see here's a drawing of glucose. That's a little bit better. So again, why are these lines fatter? They're trying to show it sort of three-dimensionally. These are still just covalent bonds. And what you can then do with glucose is you can use it as the monomer or the building block to make other carbohydrates. So if we take these glucose rings and we stick two of them together like this, there's an oxygen that holds them together. Then we have a disaccharide because there's two sugars. This chemical reaction that occurs, you actually lose a water molecule when you do this. So you lose a water to stick two monosaccharides, simple sugars, together to make a disaccharide. This chemical reaction that occurs is called dehydration synthesis. Dehydration, to lose water. So there's not quite enough room here. Let me get rid of this for a second. Sorry, I know it's a little awkward to be switching around, but this is what we're stuck with in the time of COVID. Okay, so let me just elaborate on this a little bit more. Okay. Here's an H and an OH of 1 glucose, and we put that beside another glucose like this, and I'm not drawing all the other H's and OH's in there, I'm aware of that. Okay, so how do we get water in a dehydration synthesis? We get rid of one hydrogen from one glucose and one OH from another. Those go off to make H2O, and that's how we get this disaccharide with the oxygen left in the middle. And I believe this is maltose. This is malt sugar. If you've ever made beer, you've used maltose. Maltose is a good example of a disaccharide. Another example is lactose. And lactose you may be familiar with, especially if you're lactose intolerant, but I know you've heard of lactose. Lactose is milk sugar. And here's the thing, when you eat a disaccharide, your body typically makes enzymes which cut it into monosaccharides. And that's how it's absorbed. So a disaccharide is too big for your intestines to absorb. People that are lactose intolerant, they lack an enzyme called lactase. Not lactose, but lactase. Anytime you have a word that ends in ASE, you can bet it's an enzyme. And what this enzyme lactase does is it cuts lactose into two pieces so it can be absorbed. Well, some individuals genetically don't make this enzyme. And if you don't make this enzyme, then you can't cut the lactose in half and you can't absorb it. And it turns out that when big fat molecules stick in your gut and they're not absorbed, they tend to cause some gastrointestinal distress. And so you might have a bellyache or diarrhea or something like that. Okay, so you can bet how we might make a polysaccharide, right? Poly means many. And so what you're going to do then is you're going to stick a bunch of these together, a bunch of these glucose rings together. You're going to make many dehydration syntheses, right? Dot, dot, dot. Now we have a polysaccharide, which means many sugars. And they can be branched, which is interesting. Some examples of polysaccharides include cellulose, starch, and glycogen. And we want to talk about all three of these we want to talk about, actually. Cellulose, starch, and glycogen. So you might want to make a note of each of those. Cellulose, and we'll look at some actual pictures of these. If you look at drawings of them, they all look remarkably similar. In terms of the function of these, let's go back to glucose, maltose, and lactose. Glucose is a monosaccharide. Maltose and lactose are disaccharides, and these are polysaccharides. What's the function of glucose and what's the function of maltose and lactose? These are for short-term energy molecules. So this is what your body burns. Glucose is what your body burns for its immediate energy needs. And maltose and lactose are pretty much the same because your body makes that enzyme that basically turns them almost immediately into glucose. And so these all three really are for energy. And you know, you get a lot of energy out of eating sugar. There's a lot of calories in it and energy associated with it. So what about these, though? That would be the question. What about polysaccharides? Well, depends on the polysaccharide. Now we're going to see a different function for cellulose. It is a structural polysaccharide. And it is found in the cell walls of plants. The cell walls are made of cellulose, which is a structural polysaccharide. Turns out you can't digest cellulose. Your body doesn't make the enzyme which allows you to digest cellulose when you eat it. And so some people wonder, well, why should we even eat it, right? It's the fiber of plants. You've probably heard it's good to eat fiber. It's good for you. Why is it good for you? Well, it turns out that although we can't use the cellulose directly, there are microbes in your gut, the so-called flora, right, which are bacteria, that do make enzymes that cut this up, and they are able to use it for energy. And it turns out that the waste products of your intestinal flora, the bacteria that live in your intestinal gut, you can't live without them. You need them. Their waste products are things like niacin, biotin, right? These are micronutrients to us, vitamin K. These are all things that bacteria make, and they give us in this symbiotic relationship. In return, we're supposed to feed them the cellulose, right? So you got to eat plants, and you got to eat plant cell walls. You should definitely eat them. The best way I can describe cellulose, you've all eaten celery, or most of you have. I have occasionally had students that are like, nope, never eaten it. When you eat celery, and you take a big bite of it and you chew it up and suck all the juice out, the cellulose is that stuff you're kind of left with in your mouth, and you're not sure if you should swallow it or kindly spit it out, right? Swallow it, it's good for you, okay? What about starch and glycogen as polysaccharides? Starch is the storage polysaccharide, not structural, but storage polysaccharide in plants. And you know this, right? Where do you get starch? You get it from plants. You eat plants, and for example, a baked potato or pasta, you're eating plant starch. And your body takes that starch and chops it up into glucose, okay? Glycogen is the same thing in animals. It's the storage polysaccharide for animals. Here's the crazy thing about your liver. Your liver is like a chemical magician. It can turn basically any organic compound into glucose. And so when you eat starch or when you eat glucose, let's say you were late to class one day, back when we got to actually go to class, and all you had time for was to scarf down a Snicker bar and chug a 22-ounce Mountain Dew, right? What happens to your blood sugar level? It skyrockets, right? You have all this simple sugar gets absorbed straight into your blood. The liver filters it out, sticks it together, almost like Legos or Lincoln Logs or something, sticks them all together and makes glycogen where it's stored. And then let's say you didn't eat anything for the rest of the afternoon and your blood sugar level starts to plummet. Well, then the liver takes that glycogen and chops it up back into glucose, dumps it back into the blood. So your liver is constantly regulating blood sugar level. It's either putting it in or it's taking it out. Now we know that some individuals can't regulate their blood sugar level. The mechanism is broken down. We call this diabetes, right? And if you have diabetes, then you're forced to take insulin or figure out some way to treat the fact that your body can't regulate its blood sugar. So to be clear, when you eat glucose or starch, well, let's think about what happens when you eat starch. When you eat starch, it gets chopped up into monosaccharides and absorbed through the gut and into the blood, and it gets filtered out by the liver and turned into glycogen. So it can go from starch to glycogen. Okay? Good. This is all good stuff. I want to show you some images of, here's part of the cellulose molecule, and you can see we have 3 of the many rings that would be present. But let me go online just for a second. Let's look at polysaccharide images, because they'll probably show a bunch of them. You know, like I said, you'd kind of be hard pressed maybe to tell them apart. Let's see what this one looks like. So here's, oh, this is nice. This shows starch. Let's see if we can get that to get big. Yeah, so here's, oh, jumping all over the place now. I got a Walgreens ad. That's okay. So here's a monosaccharide, glucose. Disaccharide, sucrose. We didn't talk about sucrose, but that's another disaccharide. That's table sugar, right? And then here's a polysaccharide. This is starch. And so you can see 1 and then 2 for di and then many for poly. Okay. Now I've lost my PowerPoint, of course. Got too many things going on at once. Sorry about that. A lot of screens to deal with. Here we go, we're back. So, that's pretty good, I think, for carbohydrates. We say, okay, what are they made out of? Well, they're made out of carbon, hydrogen, and oxygen, right? Those are the only 3 things you see there. They can be monosaccharides, disaccharides, or polysaccharides. Their function can be structural in the case of cellulose, or in the case of glucose or starch or glycogen, they can be energy molecules. They can be immediate energy like glucose, or they can be a way to store energy like starch and glycogen, intermediate energy storage. Okay. Next, we have lipids. Lipids include fats, oils, waxes, and steroids. We often talk about monomers. Monomers are like the building blocks of the organic compound. So you could kind of think about the glucose ring as a monomer of carbohydrates. It's a little less clear when you're talking about lipids, but we'll see what they're made out of. And there's some variation in terms of what they're made out of. However, there's one property of lipids that holds true for all of these things: fats, oils, waxes, steroids. And the one thing that holds true is that they are hydrophobic. Think about what that's saying, hydrophobic. literally means scared of water, right? And what we're getting at, of course, they're not scared of water, but they won't dissolve in water. They're not miscible, or they won't go into solution in water. And you know this, the oil will float on the surface, right? It won't go into solution. Water is very polar and fat is very non-polar. And so they don't mix, okay? We have fats, oils, waxes, and steroids. Primarily, we're going to be talking about fats and oils. Here's the thing. What's the difference between a fat and an oil? Structurally, not much. Sort of arbitrarily, a fat is solid at room temperature, and an oil is liquid. So, you know, I have coconut oil here, which is what you call it, but realistically, it's coconut fat, right? Because coconut, you've seen, is solid is solid at room temperature. Think about the rim of fat around your ribeye steak compared to olive oil. Okay. Other than that, structurally they're the same. Let's talk about the structure, and then we'll talk about the function. Basic lipid structure, specifically fats and oils. There's two parts, there's two monomers. There's a glycerol molecule. And if you look, you can see that the glycerol molecule consists of 3 carbons, 1, 2, 3, along with a bunch of hydrogens and hydroxyls. Okay, so that's one of the building blocks. This is a fatty acid. And so what it consists of really is a glycerol and 3, 1, 2, 3 fatty acid. We sometimes say fatty acid tails. They're hydrocarbon chains. They're just carbon and hydrogen. That's all they are. Okay. And so there's a whole bunch of words associated with this stuff that we want to go into. Talk about okay. So let me get a fresh screen, and I'm going to just draw this out for you one time. What you're dealing with when you're dealing with a lipid is you have the glycerol. Right? And then you have these carbon to carbon bonds, like this. And we'll just make them short, and then you have H's. I'm just going to draw this one like this. Here's the glycerol. And these are the fatty acids. Okay, so here's the thing, y'all. Remember the rule about sharing electrons. Remember, each of these lines represents a covalent bond. Carbon needs 8 electrons in its outermost shell. Well, 2, 4, 6, 8. So that carbon's good. You'll notice that all these carbons have 8 electrons. What about hydrogens? They need 2. They only have one. They only have one orbital. 1, 2, 1, 2. So you get the idea. Okay. So there are all sorts of terms associated with these fats and oils. One is, for example, saturated versus unsaturated. And if you recall, one is supposed to be good for you and one is supposed to be bad for you. They've kind of debunked that to a certain extent. But what do they mean by saturated and unsaturated? Saturated with hydrogens is really what it means, or unsaturated with hydrogens. If you look at this fatty acid, you can see every carbon has 2 hydrogens attached to it. But something that can happen is these carbons can share 2 electrons. 1, 2, 3, 4. They can share 2 pairs of electrons. They can form, this is called a double covalent bond. Now, if you do that, look what happens. Now you got 1, 2, 3, 4, 5, 6, 7, 8. That carbon's good. Same thing with this carbon. 1, 2, 3, 4, 5, 6, 7, 8. That carbon's good. So this fatty acid is unsaturated with hydrogens because of this double bond. And you can have multiple double bonds or none at all in a particular fatty acid, and that determines if it's unsaturated or saturated. When they talk about mono unsaturated versus di unsaturated, right? They're talking about how many of these double bonds there might be in a given fatty acid. What does the food industry do? Hydrogenated oil, right? Oh, they take something that's relatively good for you, this unsaturated oil, and they, in a laboratory environment, force these hydrogens on like this. And so they're hydrogenating the fatty acid. Why would they do this? Almost always they do it because of money. You know, a couple of reasons. It makes it more stable, the shelf life more stable. Hydrogenated oil is shelf stable. A Twinkie can sit on the shelf for 5 years and still be a Twinkie because of the hydrogenation of the oil in it. It also tastes really good to us. It's very buttery in flavor, and so we like the way it tastes, and so taste would be another reason. It's also able to be heated up at a higher temperature, but I will tell you, hydrogenated oil, there's no safe amount. It is not good for you. There's no safe amount of hydrogenated oil in food. Hydrogenated oils now have to be labeled in food, which is nice. And I just want to tell you that one of the things the food industry will do is they will manipulate the serving size in order for them to be able to say it has zero grams of hydrogenated oil. If it has 0.99 grams of hydrogenated oil per serving, then they don't have to label it as having hydrogenated oil. So in certain countries in Europe, hydrogenated oil is banned. That's how bad it is for you. It sticks to the inside of your blood vessels and clogs your arteries and causes heart disease. So be aware what the food industry does. Okay. Now, as far as waxes go, they have a pretty complicated structure, and we're not going to really worry too much about them. We should say, what is the function of fats and oils? Fats and oils are for long-term energy storage, right? So when you eat an excess of carbohydrate, your liver can ultimately turn it into fat and store it for a long period of time. If you consume fat and you're not burning all of the carbohydrates that you're eating, then the fat just gets stored. This is one of the fundamental reasons why the diet, the caveman diet where you starve yourself of carbohydrates is effective as a weight loss, because your body, when you starve yourself of carbohydrates, burns the fat. I'm not saying it's necessarily a healthy diet, but it is an effective way to lose weight. I am not a dietitian. What about steroids? Well, steroids are hydrophobic or fat-based molecules that we find in living things. And the function of steroids are chemical messengers. They have some use as in a messaging system. Essentially, they are hormones, right? When hormones are released in the body, they have some effect in some other part of the body, usually via the bloodstream. And so the most important hormones end up being cholesterol, which is the precursor to the sex hormones, testosterone, estrogen and progesterone. As you know, testosterone is the male sex hormone. Estrogen and progesterone are the female sex hormones. Guess what? They all come from cholesterol. Now, you hear a lot of stuff about cholesterol. It's bad for you. You shouldn't eat it, etc. Your body makes cholesterol, and you can't live without it. So your body has to have cholesterol in order to survive. And one of the reasons is because it uses it to make these hormones that are necessary. And you want to think about what they do. Well, what they do is they regulate the sexual cycles of men and women. When you think about testosterone, you'll notice the structure. We might just for a second talk about the structure. You have these 6 carbon rings and then a 5 carbon ring. You'll notice it looks like a pitcher's mound. Keep in mind that the carbons are understood. You know, everywhere there's 2 lines coming together in this drawing, right here, right here, right here, there's a carbon in all those spots. Okay. So, you know, do I want you to be able to draw this? No, you don't have to be able to draw it, but you should be able to recognize it because it's very distinctive. So take a mental picture of the way this is shaped and then you'll remember. Okay. Testosterone is what makes males male. What are some male traits? Muscle mass, right? Typically, males have greater muscle mass than females. They can exhibit aggression. They typically have more hair, like facial hair. These are all things associated with men that are produced by the action of testosterone. Now, one of the things I'll point out is that from cholesterol to testosterone and then from testosterone to estrogen. So females also make testosterone. It's just that they then convert it into estrogen. Okay. Estrogen and progesterone are what regulate the female uterine and ovarian cycle. So estrogen regulates the ovarian cycle and progesterone the uterine cycle. Different levels of these hormones cause the ovary to produce an egg roughly once a month, every other ovary. And the influence of progesterone causes a buildup of the uterine lining, right? When you take birth control, estradiol is just a synthetic form of estrogen. You're tricking your body into thinking it's pregnant as a female. And as long as your body thinks it's pregnant, it won't produce, because of the high levels of estrogen, it won't produce an egg. And then on the 28th day, you take your last pill and your estrogen level plummets. That causes the sloughing off of the uterine lining and you have your period. Okay. So this is a way to control pregnancy. Okay. There are synthetic forms of sex hormones like estradiol. There's also anabolic steroids, right, which are used as performance enhancing drugs. They are illegal. They are classified as illegal drugs. You cannot possess them, buy them, sell them. But there's a huge market for them because of the fact that they can be used to build muscle mass. They can be used, you know, to enhance performance of athletes. Is there a legitimate use for anabolic steroids? Of course. If you are transitioning, for example, if you're transitioning from female to male, then you might take anabolic testosterone to facilitate that transition. It's also used to treat inflammation. Prednisone is a good example. Some of you may have taken prednisone in response to, prednisone is very similar to testosterone, and you may have taken it in response to some sort of inflammation that you may have had. Okay. Let's see how we're doing on time. 36 minutes. Okay, I think we can continue on. So I guess the point is with steroids and anabolic steroids, there is a place for them, but they have a high potential for abuse, and if you don't use them properly, they can have some serious negative side effects, especially testosterone in males, because if you take too much testosterone as a male, the excess is then converted. Your body can only use a certain amount, and the excess is then converted to estrogen. So you're actually doing just the opposite of what you are trying to do. You're then bombarding your body with female sex hormone. And this can cause things like breast growth and the shriveling of the testes. And neither of those things sound good to me as a male. So be aware, there are dangers of anabolic steroids. They can also be very dangerous to the liver and the kidneys. And they're illegal. Okay. Next, we get to proteins. Proteins can be structural or act as enzymes, and they are the monomer, or the building block of a protein, is called an amino acid. Some examples of proteins include lactase, which we already talked about. It's the milk sugar enzyme, the one that cuts the disaccharide lactose in half. And collagen is a good example of a structural protein. Collagen is found in the connective tissue that sticks your skin to your underlying muscle. Okay. So let's look at the building block. Let's look at an amino acid. Okay. And the structure of proteins gets very complicated. We're going to break it down and try to simplify it a little bit. An amino acid consists of a carbon. Oh, I don't know how it turned red there. Let's see what happened. That's okay. We have a carbon. We have something called an R group. We'll talk about what that is. We have a hydrogen. We have a carboxyl, COOH, and we have an amine. That's where the name amino acid comes from. Amine is, I believe, NH3. I'm going to have to look and see if that's right. I think it is right. Okay. This is called a carboxyl. These are functional groups. You know, in chemistry, we talk about functional groups, and carboxyl is a functional group. It gives properties to a molecule or a compound. And amine is another functional group. Now, one of the things you'll notice, this is the amino acid basic structure. And the R group is what changes from one amino acid to the next. And there are 20 different R groups, which means that there are 20 different amino acids, at least essential amino acids. And so, you know, you can change the R group, and that makes one amino acid different from the next. How do you make a protein? You take the amino acids and you stick them together in a chain like this, times 1,000 or more. It's kind of arbitrary, but we say if it's 1,000 or more amino acids stuck together, then technically it's a protein. Okay. These bonds are important, and they're called peptide bonds. Peptide bond is the bond that holds two amino acids together. So let's look maybe for a second at some different R groups. You could imagine the simplest R group would just be a hydrogen. atom, just an H, but they can get quite complicated. And let's make sure that an amine is NH3. Yes, NH3, you can see it right here. Let's blow this up a little bit. And you can see there's the central carbon, there's the R group, there's the amine, the hydrogen, and the carboxyl right there. We're missing part of the carboxyl because that's what holds the adjacent amino acids together. So one of the things is, the sequence of amino acids is called the primary structure of the protein. But then it gets really complicated. And I don't expect you to really understand all of this, but there's a secondary structure where we have these hydrogen bonds start to form, and it sort of makes these folds, and then it folds even further into what they call a tertiary structure. And then, heck, they even tell us that there's a quaternary, a fourth dimension to the shape of a protein. Essentially, what you end up with is this big, long string of amino acids that's sort of all wrapped around itself into a big ball, okay? We might just, oops. At the end, we'll go, when we're done with this PowerPoint, we'll go and look at some different R groups, and you could imagine. Now, here's the thing. Primary structure of an amino acid, or of a protein, is the sequence of amino acids. Now, if there's 20 different amino acids, and you're sticking 1,000 of them together, right? 20 amino acids, and you're sticking 1,000 of them together in a line. What are the chances that any two proteins are the same if you have 1,000 of them stuck together? Imagine if you had a refrigerator box with 20 different beads in it that you could stick together, 20 different colored beads. And I randomly said, okay, I want everyone to make a protein. You start sticking these beads together by reaching into the refrigerator box and pulling one out and sticking it to the next one. What are the chances any two of you make the same one? That would be the question. And the answer to that mathematically is 20 to the 1,000th power, because you have 20 different things and you're sticking 1,000 of them together. That's a really big number. If you punch that into your calculator, you're going to get an E for error because it's too big of a number to calculate. So the potential for different kinds of proteins is astounding. And in nature, there are so many different kinds of proteins. Okay. We might just take a second to talk about what it is that proteins do. And they are structural. For example, collagen is a structural protein. Grab your skin on your forearm and pull it really hard. Do it right now. Grab your skin on your forearm and pull it really hard. And then let it go. What does it do? It snaps back, right? It snaps back because there is collagen in that tissue. And the collagen is like elastic. Unfortunately, as we age, our collagen starts to deteriorate and the cells that make it start to deteriorate. And so we lose the collagen over time. It's kind of sad. We start to sag. And so one of the things that they might try to do is sell you some kind of collagen cream, which is kind of a waste of money because collagen is a giant molecule. It's not going to pass through your skin and get into the underlying connective tissue. Collagen injections work where they actually take cells from your body, extract the collagen, and then inject them. And that's one of the ways they can puff your lips out or fill in your chin. This is so-called fillers, right? But those aren't actually in the cells. They're just in the spaces around cells. And so over time, your body reabsorbs them and you would have to continue to do it. Actin and myosin are structural proteins found in muscles. right? And then the other function of proteins are as enzymes. And enzymes are referred to as chemical catalysts. What does that mean? It means they speed up chemical reactions. And when they do studies about enzymes and how much faster the reaction occurs in the presence of the enzyme, 10 to the 7 times faster. And so what that means is if there's some product that you're waiting on, in the presence of the enzyme, that product is produced 10 to the 7 times faster than without the enzyme. And that just means that you can't live without enzymes. If your body heats up too much, it destroys the shape of the enzyme, and then you no longer get the product, and you die ultimately. The primary reason why you die if your temperature gets too high is because of the destruction of these enzymes. Let's talk for a second about how enzymes work. They have a very distinct three-dimensional shape, and there are notches. We'll let this be enzyme A. The way that it works is the reactants, what we call the substrate, is a particular shape. We'll let that be substrate A. And we'll let this be substrate B. Okay, and so an enzyme is very specific to a specific substrate. And what then happens is the substrate fits the active site. It fits down in here, like so. This little notch is called the active site. And a single enzyme is covered with these. I've only drawn one. And then what happens is out comes the product, which is chemically altered from the substrate. So think about what I'm saying. This enzyme catalyzes the reaction. The substrate molecule fits in there. Chemical reaction occurs. It pops out as the product. The next substrate molecule fits in. Reaction occurs. Out comes the product. The enzyme can be used over and over and produce product. Okay. One of the things that can happen, which is interesting, is that there could be a molecule that is a similar shape to the substrate. Typically, we call that an inhibitor. And inhibitors are very dangerous to living things because what can happen in the case of an inhibitor, now there's competition for this active site. I might point out there's nothing guiding the substrate into this little notch in the enzyme. It's just the molecules are bouncing around, and when the substrate bounces in there, the reaction occurs. But the inhibitor can also fit. The thing about the inhibitor is once it goes in, it never leaves, and it literally blocks this active site forever. Inhibitors can be very dangerous because they can stop a chemical reaction from occurring. I think that's an O. If that product is something that the organism needs to survive, then the organism dies. So one of the ways that modern herbicides work, they inhibit certain chemical reactions involved in photosynthesis. And so then the plant can't photosynthesize, can't make sugar, it dies. Okay? So one of the things that can happen is, which we just talked about, is the denaturation of an enzyme. What that means is the three-dimensional shape is destroyed. And then that destroys the enzyme. When the three-dimensional shape is destroyed, that active site is no longer functional. You've all denatured enzymes before, or denatured proteins at least. Every time you cook an egg, you denature the egg white, and you can see how it visibly changes, right? Okay. We're at the 50-minute point. I think I'll keep going and try to finish out organic compounds. Might run a little over our time. So just to reiterate, with proteins, they can act as structural proteins, or they can act as enzymes. The last class of organic compounds is referred to as the nucleic acids. Nucleic acids store hereditary material and are involved in protein synthesis and metabolism. And some examples that you're familiar with are DNA, RNA, and you may not be familiar with ATP, but we're going to be learning a lot about ATP here very soon. Okay. Look at the building block of the monomer of a nucleic acid of a nucleic acid is called a nucleotide. Okay, and so this is the monomer building block. And we call this a nucleotide. Okay, a nucleotide consists of 5-carbon sugar, a phosphate group, PO4 is a phosphate. There's another functional group for you. Okay. PO4, phosphorus and 4 oxygens. And then a nitrogenous base. And it's actually the nitrogenous base that changes from basically one nucleotide to the next. So this is kind of like the R group was in an amino acid. It changes. And these are the nitrogenous bases. There's adenine, there's thymine, there's guanine, and there's cytosine. And I don't know if those are spelled right. We're going to have to look them up. I'm sorry. Adenine, thymine, guanine, and cytosine. I think this is an S here. And that might be an I. I'm sorry. But you get the idea. We'll check on the spelling of these in a second. Okay, so you basically stick these things together, and that's how you end up with a nucleic acid or a nucleic acid. Okay, we're going to focus on DNA for our example of a nucleic acid. And what's cool about DNA is it is a two-sided molecule. Okay, the A stands for adenine, and the T for thymine, and the G for guanine, and the C for cytosine. So how is DNA put together? Well, you can see, think about it like a ladder. And the sides of the ladder are the sugar, phosphate, sugar, phosphate, sugar, phosphate groups. Okay, so you have those on either side. And then what forms the rungs of the ladder, here's the sugar phosphates on this side and the sugar phosphate on this side. And then the nitrogenous bases, there's one nitrogenous base on one side and one on the other. And there's a rule about the way that they stick together. If there's an A on one side, there's always a T on the other. Adenine and thymine always go together. So that means if there's a T over here, what's there going to be over here? You can always predict the other side of the DNA molecule if you know one side. It's going to be an A. Here, if we have a G for guanine, we're always going to have a C for cytosine. And then the same thing here, we have a C, we're always going to have a G. if I give you one side of the DNA, you can always predict the other. A's and T's, T's and A's, G's and C's, C's and G's, they always go together. And you can see that here in the drawing as well. Okay, and then this is nice because they're showing you a single nucleotide there. Okay, these are hydrogen bonds that hold these amino acids or these nitrogenous bases one to the other. So that's the basic structure of the DNA molecule, except it's helical. So it's kind of like if you grab the top and you grab the bottom and you twist them in opposite directions, then you get this helix, this helical shape, which is very distinctive. And you've maybe seen this in images and drawings, okay? So I think we're going to stop there, except I do want to go online now. And I want to show you some examples of R groups in amino acids. So we're backtracking a little bit. So here's a good example. Here's some amino acids. Hopefully if I click on this, I'm going to get this image. We'll see. Dun, 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 dun. Yeah, there we go. So here you go. Here are some examples of different amino acids: lysine, glutamine, aspartate, alanine, cysteine, and serine. And you can see everything stays the same. There's the amine group. There's the hydrogen. There's the carboxyl group. But look at the R groups are different one to the next. And that was what I wanted to show you. There's 20 different R groups. Okay, and then let's look up the spelling of nitrogenous bases. And you're going to see there's actually a fifth nitrogenous base that I left out. I can never remember how to spell these. I feel bad. I should just commit them to memory. Let's see. Here we go. So this even gives you the shape. Here's the fifth one that we didn't talk about yet, but we will. Adenine was spelled right. Guanine was spelled right. Thymine, I always want to put an A in there. It's just thymine. It should be easy to spell. Cytosine is S-I-N-E. Okay, so remember, A's and T's always go together, and G's and C's always go together. Okay. In terms of the function of DNA, wow, it's pretty important stuff, right? We have, as our last little bit, if we just think about DNA and what it does, stores hereditary material and passes it on, right? So you're exactly half the DNA that your mother and half of your father. This is a big one. It controls metabolism of the cell and also protein synthesis. Here's the crazy thing, y'all. The sequence of bases in the DNA determines the sequence of amino acids in a protein. So the DNA is literally a recipe book. How crazy is that? A recipe book for making proteins. Wow. We're going to learn all about that in another lecture. Y'all have been awesome. And I just want to say goodbye and thank you for listening. I hope that you have a great day. I'm still gimping on a broken foot, but I'm doing my best. Yep. All right, y'all. Take care. Bye-bye.